al Seminario de Laboratorio. El día de hoy tenemos al doctor Daniel Bianchi de la Universidad de California en Los Ángeles, que nos va a presentar este trabajo sobre la corriente de California y la parte biogeoquímica. El doctor Daniel está trabajando con nosotros en un proyecto que también se desarrolla aquí en el IPS, son estos proyectos del GI, junto con un colega de él de la Universidad de Santa Bárbara. Y bueno, pues le pedí que si nos podía dar una presentación de sus trabajos en la corriente California. Entonces, bueno, Daniel Bianchi is an associate professor of oceanography at the University of California. His research focuses on the interaction between ocean circulation, chemistry, and marine life, and is based on a combination of observations and numerical models. In this talk, he will synthesize results from multi-scale simulations with a physical biochemical ocean model to examine the biochemical dynamics of the California current system. Bueno, sin más preámbulo, Daniel, please.
Muchas gracias. Encantado. Voy a hablar en inglés.
Yes, please.
No está tan bien. Y me confundo con el italiano también. Así que mejor hable en inglés. I don't think I can talk science in Spanish yet. So
no problem.
From here in English. So yeah, thank you so much for inviting me to give a presentation about our work. I should say this is really, I'm going to present a summary, like very short summary of many results from multiple people at UCLA. Most of them are listed here over many years. So this is really the build-up of a modeling system to study natural variability, but also local effects. And a lot of the figures I'm going to show are from McWilliams' group, Pierre Damien, who's now at UCLA, previously was a researcher in Mexico in Ensenada, and many other people from my group and from local agencies and other universities. So just to get us started, we all know about ocean change. The ocean is undergoing many changes under human pressures, warming, loss of oxygen. Here is the trend from the IPCC telling us what percent oxygen the ocean has already lost, so a few percent up to 2000, and projected to decline into the future. So this is a global projected change. Acidification is another consequence of the atmospheric CO2 that has been increasing, but also coastal pollution and eutrophication and loss of organisms and habitat following fisheries and other impacts. And this is another example of ocean acidification, again, decline up to today and into the future. These changes are global, but there are also a lot of influences that are local, and I would argue that many of the impacts that we care about are really a local scale. So we're going to go from very large scales to smaller and smaller scales to be able to discuss impacts that matter for ecosystems. Probably the California current doesn't need any introduction. Really goes from Canada up here in the north. Is a broad current that circulates from polar regions to subtropical regions across the west coast of the United States down to Baja California, and is characterized by upwelling. So again, this is a coastal process driven by the winds, by which water comes up along the continental shelf. Ekman transport moves this water offshore, and this drives up water that is rich in nutrients, low in oxygen, a little more acidic, and really drives most of the biogeochemical characteristics that we see in the California current. So this is a little bit the system that we're gonna, that our work is centered on. Because of natural upwelling and because of the strong gradients between the surface and the subsurface ocean, there is a lot of natural variability on the order of days to weeks, but even longer time scales. So this is something also that we strive to capture at multiple scales, because it's really the background for anthropogenic changes to be superimposed on. really what we want to do is go from the global scales of anthropogenic impacts to the very small scales along the coast for different communities and different ecosystems. Natural variability as manifestation on all these scales, going from climatic trends to add these some mesoscale currents and fine scale mixing. Human drivers also go from global scales to local scales. Imagine, for example, pollution through river or wastewater outfalls added in all along the coast. And eventually the impacts in the California current are also a mix of all these scales. And we're going to focus really on biogeochemical cycles, so primary production, oxygen loss, acidification, and maybe some of the more local manifestation of these. So for the rest of the talk, I'm gonna tell you about the development of a physical biogeochemical model for the US West Coast. I'm gonna more or less say that physics control the very large scale variability in this system. So it's really a, if you wish, bottom-up control system in terms of physics and biogeochemistry. The continental shelf is where we observe most of this variability. There are really intensified hotspots of hypoxia and acidification that develop because of enhanced cycling in the right next to the coast in very shallow waters. If you want to model that, you need a model that captures these small scales. And finally, at these scales on the continental shelf, human impacts are important, and we'll see that pollution drives local eutrophication hotspots, which make oxygen loss worse and also drive additional acidification on top of the global trends. And by the way, if you guys want to stop me anytime, go ahead, ask questions. I'm happy to be interrupted if you want. Okay, so let's start a little bit with the model development. So our model is developed at UCLA. It consists of a regional ocean modeling system, ROMS, that is the physical component, coupled to the biogeochemical elemental cycling model, that is the biogeochemical component. We have a downscaling strategy by which we nest smaller and smaller scale simulation into larger scale. So we start with a Pacific scale at 10 to 12 kilometers. And then we have a simulation for the entire coast at 4 kilometers. And say the entire coast, but it's mostly the US West Coast. We're missing a little bit of Baja California. So our next generation of simulation will also extend further down. We also have two twin nested simulation shown here at 1 kilometer that provide a almost seamless high resolution simulation for the entire coast. All of these is run with very realistic atmospheric forcing that also come from a model. I'm going to tell you a little more about that in a minute. This is integrated for 20 years or so. We're also extending a little bit the simulation, but most of the results will cover the 1997 to 2017 period. And finally, we also have even higher resolution simulation at 300 meters. I'm going to mostly focus on a nest for the Southern California bight that encompasses Los Angeles to San Diego and Santa Barbara. And this includes anthropogenic nutrient inputs. And we are also developing now another high resolution nest in the San Francisco to Monterey Bay region. So this is our modeling system. The biogeochemical model is a typical NPZ model. So yes, multiple phytoplankton group, one zooplankton. These are coupled to cycles of nutrients. We capture nitrogen, phosphorus, iron, and silica. Of course, light drivers. This is coupled with organic matter production, both dissolve and sinking. So this transfer biogeochemical effect across the water column. There is a representation of oxygen coupled to the nutrients and the organic matter, as well as, should go back, as well as dissolved inorganic carbon. So there was a full dissolved inorganic carbon system. So this allows us to study nutrient cycles, oxygen, and carbon and acidification. Our model domain, I'm going to start talking a little bit about a large-scale simulation that we use to study the coast-wide variability. So this is 4 kilometers. To produce this simulation, we also had to run an atmospheric model that was run at 6 kilometers to really capture over the time period of simulation the variability in the atmosphere. At the time that we developed this, there were no atmospheric reanalysis that had the right resolution to drive a 4 kilometer ocean model or even finer scales. So all the model is driven by atmospheric simulation with the WRF or WRF atmospheric model. Physical boundaries come from global reanalysis, so we try to be realistic there. We interpolate biogeochemical field on density to provide boundary conditions for this large-scale simulation. And we also adapt parameterization in the model to the California and US West Coast. So we have a few adaptation in a biogeochemical parameterization to capture, for example, intensified sediment water column interaction on the continental shelf. And finally, we do this process at multiple scales. So we use the 4 kilometer simulation to drive the 1 kilometer simulation and the 1 kilometer simulation to drive the 300 meter simulation offline. So this is a one-way nesting or downscaling. All right, to talk a little bit about model analysis of variability at different scales and validation. Our first goal was ensuring that the model reproduces ocean properties, but also mechanisms. So we want the right answer for the right reasons, and mechanism is really what we want to try to capture. But also we want to try to provide a realistic representation of biogeochemistry. So we want to capture the mean state of the California current. This is a test of model realism, and most global models really fail if you go at coastal scale. So there are very large biases that we try to improve with our regional models. But we also want to test the response of the model to external forcing, natural and anthropogenic. The natural variability, we want to capture that. This happens, as I say, on multiple time scales. Depends on both local and remote drivers that we try to represent in the model. And so the way to validate and analyze the model is look at statistical properties and also property-property relationship. And we wrote a couple of papers detailing the biogeochemical and physical validation of this large-scale model. I'm going to give you a few examples of that. And the main message is really that a lot of the large-scale variability along the coast is controlled by physical drivers. So the first part is really looking at these large-scale patterns. The model more or less has realistic patterns. We think the case is that the model also captures the underlying mechanisms. Most of that is upwelling or representation of eddies and fine-scale currents. And really, there is a very strong physical-driven variability of biogeochemistry. And by the way, I should say that California is but also probably Baja California and the northern part of the domain are really great places to do model development and validation, because there is a really rich data set. And we, over the years, we put together, I would say, a very, very large data sets. These are examples of the type of observations. Most of these are from CalCoffee, but there are many other data sets. The colors show you the number of observation per grid boxes. I think these are either 25 by 25 or 50 by 50 kilometers, so hundreds of observations, both from temperature, but also nutrients, oxygen. So a really rich data set. There are many more. You can use satellite altimetry, scatterometers that tell you winds, ocean color for chlorophyll, and many other local data sets that we collected over the years. For the large scale, we mostly use the world ocean data sets, CalCoffee and satellite data. When we go to very small scale, in my group and other groups, we put together very detailed data sets that I'm going to show you some examples later on. One thing I didn't say is that, or I mentioned, is that you really want to capture the atmospheric drivers first. If you want to get a good ocean simulation at coastal regions, you have to have realistic atmospheric forces. And so Lionel Renaud in McWilliams' group over the years developed atmospheric simulations that can be used to drive an ocean model. The important thing is really capture what we call the wind drop-off. So how quickly the wind drops from the open ocean, where it tends to be stronger, to the coastal regions. If you look at data sets like the quick scat, and this is offshore distance versus wind speed, what you see is that there is a drop-off. So winds is strong, it's meridional, it's mostly southward, so here is negative. Data sets show kind of a tapering off of this drop-off, but if you run simulation with an atmospheric model, you see the winds really dropping close to zero as you are close to the coast. So you really want to capture this decline. And an example is shown in these large-scale plots. Here is how the model captures the wind drop-off and the wind stress curls. So these are data sets that underestimate that. These are other data sets. Here, so we did a comparison of this wind stress curl close to the coast and offshore. I think one of the things that we wanted to capture the most is the effect of wind changes right next to the coast. An example is the island mass effects that is particularly strong in the Southern California bight, where you have almost dipolar wind stress curl changes all around the islands. You also have effects of things like capes and the shape of the coast that also affect the winds. This affects the currents in turn. Winds is the main driver of upwelling, but also fine scale variability. So we have a few studies that show if you miss this island mass effect and the effect of capes and the coastline, you also miss some of the current features, and particularly some of the very small scale eddies that develop. So we really had to go through first downscaling an atmospheric model and then using that to drive the ocean model. And most of these will be missed by large-scale data products. In terms of physical validation, I'm not gonna bother you with all the fields that we compare, but we really look at temperature, salinity, sea surface height, eddy kinetic energy, and so forth. An example here is temperature. Temperature really tells you where the upwelling is happening. So all along the coast, you have colder temperature. This is a manifestation of upwelling. Some of the satellite products or the satellite products capture that, but they tend to potentially underestimate a little bit what happens close to the coast. So we do have a little bit of a cold model bias, especially all along the coast. We argue that it's a little bit of a model bias, but probably some of the global data sets miss the very fine scale of upwelling next to the coast. So they might underestimate the cold temperature all along the coast. But I would say the model has more or less a realistic representation of upwelling. We also compare the salinity. We have a slight bias in salinity as well. Eddy kinetic energy is realistic, very vigorous field of eddies. And we did comparison of many other variables in a paper by Lionel Renaud from a few years ago. We also did a comparison of biogeochemical data. So here is a simulation of nitrate, so the main limiting nutrient in the California coast. Generally, nitrate or iron are the limiting nutrients. I would say half of the time is nitrate, half of the time is iron. This is something we discover with our models, but is corroborated by observation. Here are the observations themselves. So again, coarse grain from the World Ocean Database. We capture realistic nutrients. There is again a strong gradient between the coast where you have upwelling, so very high nutrients all along the coast, especially in summertime, down to offshore where you have a transition to the very oligotrophic condition of the gyre. This is captured. This is compared to observation is quite realistic, but again, you probably to be more careful if you want to look at all along the shelf. You want to look at observation at smaller scales, which we did when we look at our 1 kilometer and 300 meter simulations. We also want to capture the drivers of variability in nutrient. So one thing that we look, for example, is how these variabilities, how nutrients correlate with density. There is a very strong correlation between density below the surface and nutrients at the surface. So up to almost 90% correlation between dense water and high nutrients. So whenever you have that dense water coming towards the surface, you also have high nutrients that are almost locked with the dense isopycnal. So this is the correlation in the model. It essentially tells us that as isopycnal move up and down, they bring nutrients with them, and potentially that affects upwelling waters as well. So density variability drives nutrient variability. This is also at the scale not just of upwelling, but can be internal waves. It can also be at this. So many physical processes lock density and nutrients. And this is well captured by the model. Whenever we have enough observation to look at this correlation, we have more or less similar patterns. So this is one of the mechanisms that the model captures. Nutrient variability also drives phytoplankton variability. So the first thing that we can look at is chlorophyll. So these are chlorophyll from satellites on the left, from the model on the right. We have more or less skillful simulation with a strong gradient from the coast to the offshore region. I would say maybe the model has slightly weaker gradient. So he has a little more high chlorophyll all along the coastal region, while the observations show a little more of a coastally amplified gradient. Overall, though, the transition to offshore is more or less similar. One thing that we might miss that is shown in the observation, especially in the southern part of the region, is a coastal enhancement of chlorophyll. And I'm going to get back to this feature a little bit later in the last few slides of the talk. We argue that there is an effect of pollution that is really in the first 10 to 20 kilometers along the coast. Primary production is also well captured. Remember, chlorophyll just tells you what the biomass of phytoplankton might be. It's actually not even just the biomass, but it's the pigment. So if there is a different chlorophyll to carbon ratio, phytoplankton biomass might be varying as well. Net primary production is what drives the biogeochemical cycle. And we have a much worse understanding of primary production because it's harder to observe. This is what the model produced in moles of carbon per square meter per year. We can compare that to observation. So there are many observations from Calcoffee. We have a fairly realistic representation, at least where we have these observations. We can also compare that to satellite algorithms. These are two algorithms from satellite. They vary quite a lot. Some of them show a little more coastal productivity. Some show a little less. I would say the model falls exactly in between these different data sets. So we have a more or less realistic representation of primary production. A caveat is that when we developed this model, we didn't know much about iron distribution and variability. So over the past few years in my group, we started looking a little more at the iron cycle. We put together this compilation of all iron measurements all along the coast. These are on a log scale. So there is a massive gradient between coastal region where you can have hundreds of nanomolar of iron in the water to the open ocean where iron goes down to maybe 1 nanomolar or so. The model was not really designed to capture this iron variability, but it more or less gets the gradients right. I would say it underestimates gradients. So iron is probably where I would say a lot of the biases can come from. We have a correlation with the observation. We capture more or less this coastal to open ocean gradient, but we're really starting to try to understand better what drives this variability that we observe and what is missing in the model. So I would say in the next few years, we're really going to try to understand this cycle a little bit better. And as I said, iron, half of the time, the model seems to be limited by iron. So we really want to try to understand what iron does to primary production and how it changes the variability that we observe. Primary production also is, the variability in primary production is also quite driven by physical variability. So there is a strong correlation as well between primary production and density, meaning that anytime denser water comes toward the surface, they bring nutrients and they allow higher primary production. So the correlation is about 50 to 60% all along the California current. Denser water close to the surface means higher primary production in many parts of the region. And this reflects supply of nutrients and the response of the ecosystem. And this variability is captured where we have enough observation to do this comparison, this statistical comparison. And we use here the in situ observation in the Southern California Valley. Finally, we want to talk a little bit about oxygen. This is because oxygen, of course, correlates to hypoxic waters next to the coast. So we observe generally low oxygen concentration near the coast and in the southern part of the domain. This reflects tropical water coming up, but also upwelling of low oxygen water from the subsurface. And there is this competing effect of the California current bringing high oxygen from the north. So whenever there is upwelling, there is also low oxygen. And whenever there is more intense transport from the tropics, there is also low oxygen, maybe variable on interannual time scales. These are observations, so again, realistic gradients. And the variability as well of oxygen is really tied again to the variability in physics. So you have already seen this correlation. Now it's oxygen versus density. It's a very high correlation. It's negative. When there is denser water coming up, there is also lower oxygen. And this is quite well captured by the model. You can see that in observation. You can see that in the model. Finally, the last things that we look at with this simulation is long scale trends. These are trends in oxygen concentration. So from fairly hypoxic water to fully oxygenated waters. The red is the model. There is a long-term decline. This is partly global forcing, partly I would say decadal variations like the phases of the Pacific decadal observation. More or less, we are in the range of observations. I would say maybe we get a little bit of a stronger negative trend. If we extend this to the latest year, we have a little bit of an increase again, but overall there is a declining trend. We also see a declining trend in acidification metric. This is, for example, for the saturation of calcium carbonate that is affected both by decadal variability and the global acidification trend. And we have fewer and fewer observations relative to oxygen, but the model more or less captures this long scale trend. So again, I would say there are limited data to capture trends. If you trust the model, we have both decadal variability and the anthropogenic centennial trend that are superimposed. Now I want to zoom a little bit onto the continental shelf and show you how hypoxia and acidification are really intensified on the very coastal water because of very intense biogeochemical cycling and maybe coupling with between the sediment and the water column and effects from nutrients coming to the ocean from terrestrial sources. So kind of the take-home message is that the shelf is really the biogeochemical engine of the California current biogeochemistry. Upwelling really happens on the continental shelf, and then there is a lot of transport of properties like nutrients and organic matter to offshore waters. And there is really hot spots of variability on the continental shelf. For this part, I'm going to focus on, so this is, by the way, to tell you what's the width of the shelf all along the coast. It ranges between a few tens of kilometers in the southern part of the domain up to maybe 50 to 100 kilometers. So if you want to capture the continental shelf, you really need to have a model that captures the scales of few tens of kilometers. As I said, there are strong biogeochemical cycles, and you need models that go down to sub-mesoscales. So something like 1 kilometer or even less to represent the dynamics, the currents, and the biogeochemical coupling on the continental shelf. An example here is circulation. So is the vorticity in our 4-kilometer simulation. And this is how it compares with the relative vorticity at 1 kilometer simulation. So there is really much stronger variability, more intense eddies and fronts at all scales that develop when you start going to sub-mesoscale resolving simulations. And in a few papers with McWilliam groups, we look a little bit at the biogeochemical consequences. If we focus really on what happens on the continental shelf, so now I'm going to show you this strange coordinate system where we look at the isobath. So we go from 0 to 200-meter depth, and we also look at properties on a depth isobath coordinate system. So what I'm showing here is currents. Wherever there is a red colors is a poleward current. Wherever there is a blue color is a equatorward current. So all the blue is the California current coming down. All the red is the undercurrent going up. And then the contours show you streamlines of upwelling. So in summertime, you have intense overturning that brings water up, but you also have a cell in the bottom boundary layer that transport water kind of a little bit downslope and offshore. So these are some of the properties that are really intense on the continental shelf, and especially the upwelling is really massive. Most of the upwelling that we see happens in these 0 to 200-meter depths. This is where the wind drop-off is stronger, so that's the main driver of the upwelling, and also the wind stress curl has the most variability. There is also strong seasonality, so you go from very strong summer upwelling to relatively weak upwelling in winter time. If you go north enough, this cell will flip and you will have even downwelling in the north. And again, I want to really emphasize the role of boundary layers. So there is super strong offshore transport in the Ekman layer right at the top, but there is really also a very strong offshore transport that we observe in the bottom boundary layer. And for biogeochemistry, we find that both are quite important in California. To give you a sense of this transport, I'm going to show you here the transport of nitrate on the shelf. So we're looking at the boundary between 200-meter depths, isobath, inshore from offshore. This is transport. If it's positive, is out of the shelf. If it's negative, is into the shelf. For nitrate, and by the way, this is time climatological year, so from January to December. Whenever you see a blue color, there is essentially transport into the shelf. So this is the upwelling that is mostly in the interior. Wherever you see a red color, there is transport offshore. So this is the Ekman transport in the boundary layer. The net is negative, so there is a net transport of nitrate into the continental shelf that is turned into organic matter. So there will be also a net export of organic matter offshore. But there is also a very large upwelling and transport of nitrate from the Ekman layer that will go offshore and fertilize the offshore region. So all the nutrients that we find offshore really come from this upwelling cycle and are upwelling the interior and transported offshore in this surface boundary layer. There is also a weak transport of nutrients in the bottom boundary layer for nitrate. If we look at iron, the picture is quite different. Iron is sourced mostly from the sediment, so there is essentially a net transfer of iron from the continental shelf to the open ocean. Some of that is in the surface boundary layer, but really most of that is in the bottom boundary layer. This iron goes essentially all along the shelf, crosses the 200-meter isobath, and some of that gets scooped up in eddies and potentially can even upwell once you are a little bit further offshore. So some of the processes that we're trying to understand now are really at the small scale, trying to understand the interaction between this surface and bottom boundary layer in California. I'm really kind of excited about looking at these bottom boundary layer processes. On the California shelf, also we have a development of hotspots of acidification and hypoxia. This high resolution model allows us to study them in the model, almost sample that as if it was the real world. So again, in this isobath versus depth coordinate system, we can start looking at the frequency of hypoxic events. These are events where oxygen drops below a certain threshold that might have impacts for marine organisms, so about 60 millimole per cubic meter. And we can look at different parts of the coast. In all these regions, we have hypoxia essentially developing in the upper part of the continental shelf in the north, and the central region, and deeper in the southern part of the region. And hypoxic condition might be experienced maybe 1% to 10% of the time. Acidified condition, we can also look at the same variability. With the model, we can identify hot spots. So there are several all along the coast. These are where hypoxia might reach maybe, well, if it's one, it's 100%, so it's always hypoxic. But I would say some of these regions at the bottom is maybe 10% of the year. We can also look at statistics of hypoxic events. So we can look at, for example, the duration. We can look at how strong they are. We can look at what's the return period, so how often they happen. And we can do this characterization all along the coast. And we really identify a few regions like the San Francisco and Monterey Bay. This is the shallow shelf of Oregon. And not much in the Southern California bight. You have to go a little bit deeper for that. And again, the model allows to look at statistics of hypoxia. As part of this work, we're also looking at biological impacts. This is work done by some other folks, but I'm not gonna tell you much about that in the sake of time. The really last 5 minutes or so, I want to go even at finer scales and tell you a story about pollution and how it drives eutrophication really in the Southern California bight, which in turn worsen this hypoxia and acidification. Essentially, the story is that there are local nutrient inputs that drive biogeochemical change. They reduce oxygen below the surface. They make the water a little more acidic. So we're going to focus on the Southern California bight. Whenever you see here a yellow square, this is the input from rivers. This is a region that is very seasonal. There are river inputs mostly in winter time. They're not major, but they carry nutrients both from agriculture and from anthropogenic sources. And then there is a giant urbanization. So the city of Los Angeles is here. It's massive. Santa Barbara is here. San Diego is down here. Tijuana is further down. So there are outfall that transport nutrients, mostly nitrogen. So wastewaters are treated, but they are very rich in ammonium, and they end up in the water. There are 4 major outfalls. One is in the Santa Monica Bay, 2 are in the San Pedro Bay, 1 is offshore San Diego, and then there are about 14 minor wastewater outfalls that introduce wastewater all along the coast. So we compile all these observations. This is an example from the Santa Monica Bay. The Hyperion treatment plants is essentially a pipe that is 5 mile long. It discharges nutrients with a diffuser at about 50-meter depth, and then the nutrients are kind of transported by the water circulation. All right, so we collected nutrient input specifications so that we could include them in the model. This was done in collaboration with all the local agencies that monitor pollution. We implemented these point sources in the model. We really went to the 300-meter resolution because we want to capture some of the dynamics of the plumes. And then we did a very painstaking local validation together with stakeholders, so much smaller scale focused validation of the model. And finally, we assess the effect of these wastewater effects on biogeochemistry. And again, we're trying to do some assessment of biological impacts today. also looking at harmful algal blooms, kelp dynamics, and habitat of benthic organisms. I'm going to give you a couple of vignettes. So again, this is the system. We have a natural model without outfalls, and then we have simulation with outfalls. So there is this tension between natural upwelling and nutrient inputs. These are example of our simulations, but also validation. So this is ammonia in model simulation. All the dots are observations. So I would say if you observe ammonia concentration that are above 1 or 2 micromolar, this is usually a sign of anthropogenic inputs. All along the coast, we even observe concentration up to 10 or more micromolar. So these are, or millimole per cubic meter. So these are really strong ammonium concentration. We know the outfalls, the wastewater is extremely rich in ammonia. So we attribute these to the outfalls. If we have simulation with the outfall, some of the high ammonia reaches the surface as compared to simulation without the outfalls, where we don't have these plumes of ammonia coming up to the surface. This is an example of simulation in the model. Let's see if we can start. It's a logarithmic scale, so it really kind of exaggerates a little bit the ammonium concentration, but you see really the effects of the outfalls and how quickly the water dilutes, but also transports high nutrients all along the coast. So one thing that we found is that the nutrient inputs, even though they are local, they spread all along the coast, and the effects on phytoplankton and biogeochemistry are really spread over tens, if not hundreds of kilometers. These are comparison with chlorophyll. So satellite observation, and the dots are local measurements. Here is a model with pollution, so adding the anthropogenic nutrients, and this is a model without pollution. This allows us to say that, first of all, you need pollution to, well, first of all, pollution increases the biomass of phytoplankton, here shown as the chlorophyll, but really you need anthropogenic nutrients if you want a realistic simulation of chlorophyll in the Southern California bight. If you want something realistic, you cannot run a model without anthropogenic inputs, and you really see this amplification of phytoplankton biomass all along the coast once you add the pollution. The last couple of slides, I'm going to show you the effects on biogeochemistry. So we did a careful comparison of the model with and without pollution. Here is the change in primary production in percent all along the coast, going from Santa Barbara here down to San Diego here, and essentially all along, I would say the Los Angeles coast, you see an amplification of primary production, very coastal, but up to 200%. So a doubling or a tripling of primary production in the coastal region. Again, we thought this was a very upwelling dominated, but we see an impact of local inputs all along the coast. We can also look at decline in oxygen and pH, and there is a measurable effect. Well, I say measurable. There is a detectable effect in the model. It's much tougher to measure the effect of outfalls in the observation, but if you trust the model, we can attribute an oxygen decline to coastal inputs. If you want to look at that as a function of distance from the shore, this is oxygen change. Again, comparing, taking a simulation with anthropogenic inputs minus the control without. So what we see is a decline of oxygen of several tens of millimole per cubic meter right below the surface, an increase in oxygen in the surface caused by the higher phytoplankton. But if you are a benthic organisms that lives along the bottom, or if you live subsurface, this decline in oxygen might be important. So we are trying to understand the biological consequences. These are colleagues that are working together with local agencies. And again, there are ongoing conversations with local stakeholders to think about the effects of reducing wastewater inputs and how that might affect eutrophication and potentially harmful algal bloom and other effects. All right, I'm going to stop here. I know I kind of went through many results, but I'm happy to take a few questions, and I'm gonna put the outline up here so you guys can tell me a little bit if you wanna more details on some of these results.
Thank you, Daniel. Can you listen to me?
Yes, I can hear you.
Well, thank you for your work. It's very impressive, all what you've been doing and for your current system. It's kind of overwhelming. It's just too much.
I know. I kind of wanted to show you a little bit of everything, but it's really work for many years and many people.
Yeah, I can see that. So let me see if we have questions. Well, I have some questions, maybe. So from the Calcoffee surveys, so you gather a lot of nutrients and oxygen from the surveys, I guess, right?
Yes. So a lot of the data come from Calcoffee.
So they've been taking nutrients or biochemical information since how long? You know that?
It's actually very early. So the 60s.
So since the very beginning, they thought of not just taking physical, but also biochemical information, right?
I would have to check, but yes, I think nutrients go pretty back in time. I'm trying to think a little bit because we usually, I look at the same time period of the model, and the earliest I usually go is like the 80s. But yeah, the data is public, and if anybody, in your group or there wants it, I can even pass a structure with all the observations well organized. So yeah, it goes back in time.
And how do you decide to look at iron? Also,
can you repeat?
Iron.
Yes. So
iron. What about iron? What can you say?
Yeah, I cannot hear you very well. Try again.
Can you hear me now?
Yes.
I was thinking about iron. I don't know if they've been taking
Yeah, I can tell you, you break up a little bit, so I couldn't hear perfectly. But I'll tell you a little bit. So iron is something we got interested in my group because we got a project funded with folks that go observing that. And we realized the model is very simple in its representation of iron. And it turns out there is a lot of sources from sediment, rivers, dust deposition, but it doesn't stay in the water. It's very quickly scavenged and kind of goes back into the sediment. So there is really short residence time that makes it completely different from other nutrients. And Calcoffee does not observe iron routinely as a routine measurement. So a postdoc in my group, Annie Pham, kind of put together all the studies that people had done in the last like 20 years or so, and he put together this compilation of iron. So this is a new compilation. We're trying to write a paper now and release. And we also did a comparison with the model. So there is some correlation, but I would say it's probably the weakest link in our model. I don't know how it affects the biogeochemistry. I would say if you change the iron cycle, you change everything else. So it's something we're looking into right now.
I think it's very interesting for a coastal region like here in Yucatan because we also have the upwelling.
I agree.
There are many differences, but I think I'm like very interested in looking more at your works, making your works as a starting place to see how can we develop a biochemical modeling of the Yucatan Peninsula. And I think well, you have a lot of things to show us and a lot of things to learn. So, well, thank you. I don't know.
There's a question online, I see. I can read it.
Can you read it, please?
Yeah, the question is about the boundary condition for the hydrographic model if they consider data assimilation to force the model or how the boundary conditions were made. So we don't use data assimilative models, and we don't do any data assimilation. These are really evolving simulation where essentially everything is forced at the boundaries, either top boundary condition or the lateral boundary conditions. For the lateral boundary conditions, I didn't go in detail, but we take reanalysis boundary condition from the physics. So the boundary conditions are from models that are data assimilative, not our simulation, but the boundary conditions are from a global data assimilative solution. And we impose more or less at monthly time scale. And then we also interpolate all the biogeochemical variables on density. So every time at the boundary density changes, also the biogeochemical condition changes a little bit. So we have a little bit of a coupling between biogeochemistry and physics at the boundary. So this is all methods that were developed by McWilliams group even before I joined UCLA. And we're kind of exploring, there are no global reanalysis of nutrients that I'm aware of. So we need a lot of cooking to develop these models, but there is a lot of knowledge that has been accumulated over the years that, it's written in many papers, and I'm happy if anybody's doing modeling there, I'm happy to chat about all these.
I have a question here.
Thank you, Alejandro. Very nice, Daniel. Very interesting. Amazing. I have a question. Well, I understand what I understood is that the anthropogenic input of nutrients play an important role, right? It makes a difference.
Yes.
Including them or not including them. Then in places where data is not as good, spatially and temporally as could be the northern Yucatan coast, you think we could use like the population density of the watersheds in California and make something kind of proportional?
Yeah, that's a great question. So first of all, I should say that the nutrient inputs are really important. Well, where you have big rivers. So if you go to the northern and central coast, we really need the Golden Gate from San Francisco, and we need some of the rivers. In the Southern California, but the rivers were not as important, but the outfall, so the wastewater inputs were the most important part. So everything depends on the region where you are. For California, we had a lot of data from rivers, the USGS, what's that? The Geological Survey collects nutrient measurements in rivers. So we kind of know a little bit how they vary, but they vary a lot. So some watersheds are very clean, some watersheds are very agricultural. So I would say it's very hard to extrapolate. There are some, you might be able to extrapolate for certain specific region and watersheds. The other thing that matters a lot are cross boundaries inputs. So I didn't talk much about that, but if you go to the entire Southern California coast, there are nutrients that come from the Tijuana River, and then they kind of go north along the coast. So there are what we call the transboundary inputs. So I can imagine, like in the Gulf, all the water from the Mississippi River might circulate in some way, and it might impact even the Yucatan. Maybe that's a thought. So you really need to have a sense of all these nutrient sources from many regions. I would say in some parts the natural cycles were more important than the coastal cycles. But where there is pollution or big rivers, then we had both things being important. So it's hard to extrapolate, I would say, once you get to coastal systems.
Of course. Yeah, thank you.
I think we don't have any more questions, Daniel. So thank you again for your presentation. And
yeah, thanks a lot. And I love to visit and talk in person, or hopefully with our project we'll be able to do that.
yeah, I think we can think about that in because we already have the funding for the project now for the Mexican part. So now we can really talk about the visiting to you guys and you coming to us also to visit the laboratory.
Okay.
Well,
Great.
thank you again.
Thanks a lot. Thank you guys. Thanks for coming. All right.